Okay, let's dive in. We're constantly grappling with a microscopic world around us, aren't we?
We really are.
Whether it's, keeping medical environments sterile, making sure our food is safe, or just managing cleanliness day to day. We're always trying to control microbial life,
Absolutely. And often our first thought might go to chemicals, disinfectants, antiseptics, things like that.
right?
But what happens when we need to eliminate harmful microbes without those maybe harsher ingredients? How do purely physical forces get the job done?
That's the exact question this deep dive tackles. We've pulled together sources that explore this fascinating world of physical antimicrobial agents.
Hmm.
We're going to unpack methods using heat, radiation, filtration, and even pressure to control microbial populations.
Our mission today really is to guide you through how these different physical approaches work, at a fundamental level.
Yeah.
Understand their mechanisms and explore what makes each method effective in its context, or sometimes why they maybe fall short against certain microbes.
Before we dig into the specific methods, the sources highlight some fundamental factors, things that influence how well any antimicrobial treatment works, physical or chemical.
Mhm.
Think of them as the sort of ground rules.
Exactly. The
Yeah.
sheer number of microbes you're starting with, that's critical. Higher concentration makes the job much harder.
Makes sense.
The intensity or concentration of the agent you apply is, well, obviously key. Temperature plays a significant role, even if heat isn't your primary weapon.
And how long you expose the microbes, the duration.
Oh, crucial. Absolutely crucial. It's not just about the force, but for how long you apply it.
Right.
And then there's the nature of the microbes themselves. We're going to spend some time on bacterial endospores because, well, they're notorious for resistance.
The tough ones.
They're really tough ones. But even the growth phase of a regular bacterium affects how susceptible it is.
Does the environment matter too? Like pH levels?
Yes, pH can definitely influence effectiveness. And critically, things like organic material, think blood, tissue, even just food bits, can act like a shield.
Protecting the microbes.
Exactly, protecting them from whatever method you're using. So you have to consider all these factors together.
That makes a lot of sense. So keeping those core ideas in mind, let's start with maybe the most ancient and universally understood method, heat.
Heat is foundational, yeah. We generally look at two main types, moist heat and dry heat.
Simple boiling is a form of moist heat, right? We boil water to make it safer.
Boiling at 100 degrees C, sure, it's useful. It kills many vegetative bacteria, fungi, most viruses pretty quickly.
But
But, and the sources are very clear on this, boiling alone is not a sterilization method. It just can't reliably kill bacterial endospores or even some heat-resistant viruses.
okay, so not true sterilization. For that, with moist heat, you need something more powerful. Which brings us to autoclaving.
Autoclaving, that's the gold standard for moist heat sterilization. It uses steam under pressure to get temperatures above boiling, typically 121 degrees C.
Right.
A standard cycle is often cited as 15 minutes at 121 degrees C and 100 kPa of pressure, kilopascals.
And the sources point out a really important distinction here. It's the high temperature, the 121 degrees C,
Yeah.
that actually kills the microbes,
Exactly.
not the pressure itself. The pressure is just the tool to allow the water to reach that superheated state without boiling away.
Precisely. At normal atmospheric pressure, water boils at 100 degrees C. Increase the pressure, you increase the boiling point. You need that 100 kPa partial pressure of steam to hit 121 degrees C.
Okay.
And this reveals why getting the air out of the autoclave chamber first is so critical.
Why is that?
Well, if air is trapped, its partial pressure adds to the total pressure inside. So there's less room, effectively, for the steam's partial pressure to build up.
So you won't actually reach the target temperature needed for sterilization.
You won't hit 121 degrees C if there's too much air left.
That feels like a crucial detail often overlooked. And the sources have some really eye-opening data on how the volume of liquid you're sterilizing dramatically impacts the time needed.
Oh, absolutely. It's not just the 15-minute holding time. That time only starts after the center of your material actually reaches 121 degrees C. Right. For small volumes, maybe up to 50 milliliter, heat transfer is quick. But the larger the volume, the slower it is for the center to heat up.
Gotcha.
The sources give specific examples. While 15 minutes might sterilize 200 milliliters of liquid medium once it reaches temperature, a big 10-liter glass bottle, that can take around 80 minutes just to get to 121 degrees C in the very center.
80 minutes just to reach temperature. And the holding time is on top of that.
Exactly.
Looking at the different container sizes in the table, a 125 milliliter flask might need, what, 12, 14 minutes total cycle time.
Yeah, something like that.
But a large 2,000 milliliter flask jumps to 30, 35 minutes. And a huge 9-liter bottle needs 50, 55 minutes.
Yeah.
That's a massive difference based purely on volume and how heat penetrates.
It really underscores that sterilization cycle times aren't one size fits all. They absolutely have to be validated for the specific load, the specific container size, to make sure everything gets properly heated through.
So what is moist heat actually doing to the microbes at that high temperature? How does it kill them?
Its primary mode of action is protein denaturation. The presence of water at high temperatures is really efficient at breaking the bonds, hydrogen bonds, hydrophobic interactions that hold proteins in their functional 3D shapes.
So it messes up their structure.
Totally disrupts essential enzymes, structural components. It also damages cell membranes, messes with the lipids and proteins there, and can even cause direct chemical reactions with cellular molecules because of the H plus and OH ions from water at high temp.
And that's why moist heat is generally more effective and faster than dry heat.
Exactly. Denaturation is much, much slower without significant water. So dry heat methods need higher temperatures and longer times.
Speaking of dry heat, how does that work then?
Well, dry heat methods include things like incineration, literally burning infectious waste,
Okay.
and using hot air sterilization ovens. These ovens typically run at temperatures like 160 or even 180 degrees C, but require much longer exposure times than autoclaving, often like 1.5 to 2 hours.
And the mechanism is different from moist heat.
Yeah, dry heat primarily works through oxidation. It essentially, well, it burns up the organic components of the cell, oxidizes them.
A key limitation compared to moist heat, the source notes, is penetration, right?
That's right. Dry heat penetrates materials much more slowly than steam does.
Mm-hmm.
Trying to sterilize bulky items like medical packs wrapped in cloth or something, it's far less efficient in a dry oven than in an autoclave where steam can penetrate quickly.
All right, moving from heat to another physical force, radiation.
Radiation, using energy, waves, or particles to damage microbial cells. We primarily talk about two types here, UV light and ionizing radiation.
UV light, something we're all familiar with from sunshine.
Right. The UV spectrum goes from 100 to 400 nanometers. For killing microbes, we're most interested in the UVC range. That's between 200 and 280 millimeters.
Okay.
UVA and UVB, while they're in sunlight and can damage us, they're less germicidal than UVC. The sources point out that UV below 200 millimeters needs a vacuum to travel, so it's not really relevant for typical air or water treatment.
And the germicidal sweet spot within UVC, where does it work best?
Around 260 nanometers. That's because DNA has its peak absorption of UV energy right around that wavelength.
So it targets the DNA. How does UV light actually damage the cell then?
Well, UV energy damages nucleic acids, mostly DNA, through both direct and indirect ways.
Okay.
Direct damage caused by energy between about 220 300 millimeters leads to things like photodimerizations. That's where adjacent pyrimidine bases, like two thymines next to each other on a DNA strand, get linked together abnormally, forms a dimer.
Right. I've heard of thymine dimers.
Yeah. Cytosine can also undergo photohydration. You can get cross-links between strands or bases, things like that.
And indirect damage, what's that?
That happens mostly through the generation of reactive oxygen species, ROS.
Okay.
UV energy can interact with water and other molecules in the cell, creating these highly reactive chemicals. The ROS then attack DNA, causing single-strand breaks, protein DNA cross-links, various base modifications.
Yeah.
The sources suggest that for UVA, which isn't as directly damaging to DNA, most of its killing effect is likely from these photooxidative processes from the ROS it generates.
But this isn't just a one-sided fight. Microbes have developed defenses,
Right.
particularly DNA repair mechanisms, and the sources detail some pretty sophisticated ones.
Oh, they are quite incredible. One is called photoreactivation, or the light mechanism. It's almost sci-fi.
Really?
Yeah, in the presence of visible light, specifically violet to blue wavelengths, an enzyme called photolysis can actually find those UV-induced pyrimidine dimers and use the energy from the visible light to break the dimer apart. It directly reverses the damage.
So they use one form of light to undo the damage caused by another form of light. That's amazing.
It truly is a remarkable adaptation. Then there are the dark mechanisms, which, as the name suggests, don't need visible light.
Okay.
Nucleotide excision repair is a major one. Enzymes basically scan the DNA, find the damaged bit, cut it out, and then DNA polymerase uses the undamaged opposite strand as a template to fill in the correct sequence.
Like patching it up.
Exactly. Another is post-replication recombinational repair. If the DNA copying machinery, the polymerase, hits damage and just skips over it, leaving a gap,
Hmm.
this system can fill that gap by sort of borrowing a matching section of DNA from the other copy of the chromosome, the sister chromosome, and then repairing that borrowed section later.
Clever. And the SOS error-prone repair. That sounds a bit desperate.
It kind of is. It's a stress response.
Hmm.
When the DNA damage is overwhelming, the system kicks in. Special polymerases can actually synthesize DNA across the damaged site, even though it's garbled, essentially guessing the sequence.
Risky.
Very risky. It allows replication to continue, which might save the cell's life in the short term, but it massively increases the rate of mutations.
I see.
Though the sources note that even with these dark mechanisms, eventually the cell often still needs to use excision repair to truly fix the underlying damage later on.
Wow. Despite these microbial defenses, though, UV light is used in quite a few practical ways.
Oh yeah, it's effective for disinfecting surfaces. Think labs, operating rooms, food processing plants. It's used for air purification. And it's a pretty cost-effective way to treat water without adding chemicals.
Where else?
You see it in the food and beverage industry, maybe for sterilizing syrups or treating packaging materials, even in agriculture for things like treating surfaces during meat and cheese maturation.
But it has significant limitations, right?
The biggest one is its very poor penetration. UV light gets easily absorbed by glass, plastics, even thin layers of liquid or dust.
So surface only, really.
Pretty much.
Oh.
It only effectively disinfects surfaces or very clear, shallow fluids. And of course, it's a health risk for us: eye damage, skin cancer potential.
So UV is great for surfaces in clear air or water, but for penetrating materials, you need something with more punch, like ionizing radiation.
Exactly. Ionizing radiation, X-rays, gamma rays is way up there on the energy scale. About 10,000 times more energy per photon than UV. Very short wavelength, less than 40 nanometers.
How does this much higher energy affect microbes differently?
Its main killing mechanism is by ionizing water within the cell. Water makes up a huge chunk of the cell, right?
Yeah.
This high energy knocks electrons off water molecules, creating highly reactive free radicals, especially hydroxyl radicals, OH. These things are incredibly unstable.
And dangerous.
They react instantly with almost any molecule they bump into inside the cell.
And the most critical damage is to the DNA again.
Yes. While these radicals hit everything, proteins, lipids, you name it, the most lethal damage happens when they react with DNA. They can break the sugar phosphate backbone.
Causing breaks in the strands.
Exactly. Single-strand breaks, or even worse, double-strand breaks. Double-strand breaks are really difficult, often impossible, for the cell to repair properly.
This helps explain why bacterial endospores are much more resistant to ionizing radiation than vegetative cells, doesn't it? We mentioned their resistance before.
It absolutely does. One of the key features of endospores, remember, is their incredibly low water content.
Less water, fewer targets for ionization.
Precisely. By removing most of the water, they drastically reduce the number of targets available for ionization and then subsequent free radical formation. It dampens down that primary damage mechanism.
And common sources for gamma rays used industrially.
Typically cobalt-60 or cesium-137, radioactive isotopes that emit gamma rays.
What are the major advantages of using ionizing radiation then?
High penetration is the big one. It can sterilize right through packaging. It's also very efficient, kills microbes effectively. And crucially, it's ideal for sterilizing heat-sensitive materials, things that would just melt or degrade in an autoclave.
Like plastics.
Plastics, definitely. Many pharmaceuticals, certain biological tissues for grafting, things like that.
Are there factors that influence its effectiveness?
Yeah, like other methods, things like the presence of oxygen and the growth phase of the microbes can affect the dose you need.
And practical applications are widespread.
Oh, yes. Food preservation is a major one used in many countries, WHO approved. You can use it for radiperization, which is basically sterilization for long shelf life, or regurgitation, which is more like pasteurization to extend shelf life.
Wow.
Widely used for sterilizing medical devices, especially single-use plastics, pharmaceuticals like antibiotics, vaccines, human tissue grafts, even things like wool to get rid of potential anthrax spores, and sometimes used intentionally to cause mutations in research.
Okay, so we've covered killing with heat and energy waves. What about just physically removing microbes?
That brings us neatly to filtration.
Now, the sources point out a really important nuance with filtration straight away. It's not quite sterilization in the absolute sense.
That's a critical point to understand, yeah. While filtration is excellent at removing bacteria, standard bacterial filters often have pores that are large enough for viruses and mycoplasmas, which are much smaller than bacteria to slip through. So it removes most microbial life, often reducing the load drastically, but it typically isn't guaranteed to produce a completely sterile filtrate. You might still have viruses in there.
Got it. Reduces the load, doesn't necessarily eliminate everything. What are the main types of filters?
We generally talk about depth filters and membrane filters. Depth filters are thicker materials made of a random tangle of fibers, could be cellulose, glass microfibers, things like that. They trap particles throughout the depth of the filter material. It's not just a surface effect.
Like the HEPA filters in air purifiers or labs.
Exactly. HEPA filters are a perfect example of depth filters used for air. High-efficiency particulate air filters, usually made of randomly arranged borosilicate glass fibers. Now, here's a surprising detail from the sources.
Okay.
The actual air spaces between the fibers in a HEPA filter are generally larger than 0.3 micrometers.
Wait, really? But they're rated for 0.3 milliliter particles, so they aren't just sieving them out based on size.
No, not primarily for particles around that size, which is counterintuitive. Larger particles, say above half a micron, get caught by basically running into fibers' impaction or interception.
Right.
Tiny particles, much smaller than 0.3 monometers, get caught because they wiggle around due to Brownian motion and eventually hit a fiber that's diffusion.
Okay.
But particles around 0.3 manometers are kind of in this awkward middle ground. They're too large for diffusion to be super effective and too small and light to be effectively caught by impaction interception just due to airflow. They tend to follow the air streamlines around the fibers.
So how do they get caught then, if they're the hardest size?
It's the combination of all these mechanisms working together across the depth of the filter. Even though 0.3 manometers is the most penetrating particle size, the filter is designed so that the overall efficiency at that size is still incredibly high, 99.97%. It's not just a simple sieve.
That's fascinatingly complex. Okay, what about membrane filters then?
Membrane filters are probably more common for liquid filtration in the lab. They're very thin films, usually made of polymers like cellulose acetate or polysulfone. They have very uniform pores created during the manufacturing process. These act much more like a true sieve.
And common pore sizes are designed to catch bacteria.
Yeah, typically 0.2 or 0.45 micrometers. The 0.2 mitophobia size is generally considered sterilizing grade for removing bacteria from liquids, though again, remember the virus caveat.
And you use pressure or vacuum to push the liquid through.
Yes, exactly. You either apply pressure from above or use a vacuum below to pull the fluid through the membrane. While size exclusion particles physically being too big for the holes is the main idea, other things contribute a bit too, like bridging, where particles start to build up over pore openings, effectively making them smaller, and sometimes just non-specific adsorption, particles sticking to the filter material itself.
The sources also mention nucleopore filters briefly. What's special about them?
Nucleopore filters are interesting because of how they're made. You start with a thin plastic film, often polycarbonate. Then you bombard it with nuclear radiation.
Okay.
This radiation creates tiny, straight damage tracks through the film. Then you chemically etch the film. The etching solution dissolves the plastic much faster along those damage tracks.
Creating perfectly straight, uniform pores.
Exactly. Very uniform cylindrical pores. The size is controlled really precisely by how long you etch it for. They're often used in microscopy, like for collecting samples on a flat surface to look at under an electron microscope.
Fascinating ways to make tiny holes. So we've covered heat, radiation, filtration. What's the final physical method highlighted in our sources?
That would be hydrostatic pressure, or HPP.
HPP.
You might also hear it called Pascalization, after Blaise Pascal, the scientist. Or sometimes, less commonly, Bridgmanization, after Percy Bridgman, another physicist who worked on high pressure.
Interesting namesakes. The sources describe this as a relatively new technology, at least commercially.
It is, yeah. While the science has been understood for a while, its commercial use, especially in food processing, really only started taking off in the 1990s. It's still considered somewhat of an evolving technology.
How does it work? What kind of pressure are we talking about here?
We're talking very high pressure. To knock out vegetative bacteria, molds, yeasts, and some enzymes, typical pressures are around, say, 340 megapascals. That's roughly 3,400 times normal atmospheric pressure.
Wow.
Applied for maybe 15 minutes or so.
That's an immense amount of pressure. How do they kill a microbe? What's the mechanism?
The primary mechanism seems to involve disrupting non-covalent bonds. Things like ionic bonds, hydrogen bonds, hydrophobic interactions.
The bonds that hold proteins together.
Exactly. These linkages are absolutely essential for keeping proteins folded in their correct functional 3D shapes. High pressure messes with these bonds, causing proteins to denature, to unfold. The extent can vary depending on the protein. It also significantly disrupts microbial cell membranes.
And are those tough bacterial endospores resistant to HPP too?
Uh,
Seems like they resist everything.
Yes, they are a major challenge for HPP, just like they are for heat and radiation. Endospores can tolerate incredibly high pressure, sometimes up to 1200 MPa.
Whoa.
So while HEP is very effective against vegetative cells, it's much less effective against endospores. They often survive standard HPP treatments used in food processing.
The sources mentioned some newer research combining pressure and heat. Trying to tackle that.
Right. There's work being done combining more moderate pressure, maybe around 0.14 MPa, so much lower, but combining it with heating to maybe 82, 103 degrees C. This combination seems to enhance the killing effect, potentially improving shelf life by tackling some enzymes or more resistant spore forms that pressure or heat alone might miss. Still developing, though.
How is the HPP process actually carried out, like industrially?
It's typically a batch process. The product, often already packaged, is loaded into a big, strong, high-pressure vessel.
Packaged already.
Yeah, that's a key advantage. The vessel is then filled with a fluid, usually water, which transmits the pressure. Pumps then raise the pressure in the water up to the target level and hold it there for the specified time.
And the packaging and the food inside, they don't just get squashed flat by that incredible pressure.
That's one of the really neat things about hydrostatic pressure. The pressure is applied equally from all sides.
Okay.
So instead of being crushed in one direction, the package and its contents are just uniformly compressed slightly. When the pressure is released, they pretty much return to their original shape, usually undamaged. It's quite clever.
Does HPP work for all types of food?
It works well for both solid and liquid products. It seems particularly effective or maybe advantageous for acidic foods. Things like juices, yogurts, fruit products.
Why acidic food specifically?
Because even if some of those super pressure-resistant endospores survive the HPP treatment itself, they generally can't germinate and grow out in a low pH acidic environment anyway, so the acidity provides an extra hurdle.
What are some of the overall pros and cons of HPP then?
Okay, well, on the downside, the equipment is expensive, high capital cost. And as we've said, bacterial endospores are difficult to kill with pressure alone, which limits its application for achieving full sterility in low-acid foods.
Right.
But the advantages are significant. The processing time is largely independent of the product volume or size, unlike heat where penetration takes time.
Big difference there.
Huge difference. There are no penetration problems in bulk packaging because the pressure is uniform throughout instantly. It generally has minimal negative effects on the sensory qualities, flavor, texture, color, and the nutritional value of the food. Often preserves freshness and vitamins better than heat treatment.
That's a big plus for food.
Definitely. And interestingly, it's often perceived by consumers as a more natural process because there are no chemical additives, no radiation involved. It just uses pressure. Plus, the sources note a recent finding that HPP can even kill foodborne pathogens that have managed to get inside fresh vegetables, which is a really tough challenge for surface treatments.
Okay, wow. So we've covered heat, radiation, filtration, and high-pressure processing. Let's try and bring it all together now and circle back to those initial factors influencing efficacy, particularly microbial resistance.
Yeah, looking at the comparative resistance table the sources provide is truly revealing. It just hammers home the vast differences in susceptibility between different types of microbes and even different forms of the same microbe when you hit them with various agents.
The numbers showing bacterial endospore resistance compared to something like E. coli are genuinely astounding.
They really put it into perspective, don't they? If you take E. coli, a common vegetative bacterium, as your baseline, give it a relative resistance of 1, then bacterial endospores show a relative resistance of maybe 3 million to moist heat.
3 million times harder to kill with moist heat.
Yeah. And against something like formaldehyde, a chemical agent, it's a staggering 100 million times more resistant. Even against dry heat, they're still around 1,000 times more resistant than E. coli. In comparison, fungal spores and typical viruses, while tougher than vegetative bacteria, are generally far, far less resistant than endospores to these really harsh physical and chemical treatments.
So the endospore is clearly the heavyweight champion of microbial survival.
Right.
We touched on it, but why exactly is it so incredibly resistant, especially to heat? Is it just that tough outer coat protecting everything?
That's a common assumption, but the sources clarify that the endospore coat's main job is not heat resistance. Its incredible heat resistance comes primarily from what's going on inside the core of the spore.
Okay.
During spore formation, there's this process called cortex contraction, which actively squeezes most of the water out of the central cytoplasm, the core. It becomes extremely dehydrated.
Ah, low water again. We saw that helped with ionizing radiation resistance too.
Exactly. It's key. Remember how moist heat works? By denaturing proteins, which needs water to break hydrogen bonds. In the endospore's super dry core, protein denaturation happens incredibly slowly. You need very high temperatures for a long time to make it happen. On top of that, the core accumulates huge concentrations of calcium ions complexed with dipicolinic acid. And these special small acid-soluble proteins, say SPs, together, they form this sort of protective gel-like matrix that further stabilizes the DNA and proteins against heat damage.
So the heat resistance is mainly about being incredibly dry inside, plus this internal stabilization matrix. What about chemical resistance that you mentioned, that 100 million times resistance to formaldehyde?
That's where the outer layers, particularly the endospore coat, play the crucial role. The coat is a complex, multi-layered structure, and it's highly impermeable to many chemicals.
So the coat acts like armor against chemicals.
Pretty much. It acts as a barrier, preventing things like disinfectants from even reaching the vulnerable core components inside the spore.
That's a really key distinction then. Heat resistance is mostly internal structure and dehydration, while chemical resistance is largely about that impermeable outer coat.
Yeah.
It's just a marvel of natural engineering designed purely for survival.
It truly is, and it perfectly highlights why no single antimicrobial method works for everything, and why understanding the specific microbial target, especially if it might be spores, is absolutely paramount when choosing and applying a control method.
Well, you've now taken a deep dive with us into this diverse world of physical antimicrobial agents, exploring everything from the brute force effectiveness of heat to the specific energy of radiation, the mechanical removal by filtration, and these modern high-pressure techniques.
And as we've seen, each method has its specific niche, its unique strengths, and yeah, its limitations too. Their effectiveness is so intricately linked to the conditions of use, temperature, time, concentration.
And fundamentally to the characteristics of the microbes themselves, especially the remarkable resilience we see in structures like those bacterial endospores. Right. Considering just how sophisticated microbes are, whether it's their whole toolkit of DNA repair mechanisms fighting back against radiation damage, or the incredibly complex multi-layered survival strategy packed into an endospore that just shrugs off incredible heat and chemicals.
It really leaves you pondering, doesn't it?
Are these physical methods we use just sort of chapters in this ancient, ongoing, and ever-evolving battle against microbial life?
That's a great way to put it. It's a constant arms race.
Something to think about next time you see an autoclave humming away, or trust a water filter, perhaps.